Yeah, thanks Veronique for the introduction, and yes, and so one thing I want to say is it's actually not a job requirement to run a marathon for the Fox Foundation, but I will say I ran the marathon for the Fox, I think what's interesting about working at the Fox Foundation is there's this, I work on the research team, I'm a scientist by training, I work with one of the 7 PhDs, but independent of that, we're also a family, I think, and we all sort of really get into what we're trying to do. We're all part of the same sort of ultimate goal here, and so I'm always impressed when I see so many people from the Fox Foundation staff members who get involved in Team Fox in addition to their day jobs. So I just want to give a shout out to that. All right, so I have the, I guess, enviable job today of trying to give you sort of a broad overview of the Parkinson's research field and sort of the way, really through the lens that we see it. And kind of talk a little bit about some of the challenges that we see, but also, as has been alluded to, some of the excitement that we're seeing as well. So just a few things. You all have copies of the slides in front of you. I'm probably not going to be able to go through every single point on every single slide because I think we would be here for about 3 days. So obviously take these home with you, jot notes down. It's a good opportunity to kind of educate yourself. If you have specific questions, certainly at the end we can address those, but I'm going to try to give a somewhat fast, rapid overview of all of these different areas. Another point, I don't think it's been mentioned yet, at the end we definitely want to hear from you and your questions, so to make that sort of more effective, what we're going to ask people to do is write down your questions. I think there's some note cards on each table, so if you could write down questions as we go along, you can just kind of keep them in your mind. At the end we'll actually pick those up, and then we're going to try to answer as many of them as we can with our panel. So, without further ado, we've talked about this idea of a pipeline a couple of times now already, and I think it's a good place to start because this really kind of defines the challenge that we're focused with right now, which is how do we get things from all the way over on the left side, which is really where the ideas about what causes Parkinson's come from, how we might be able to treat it. How do we get them actually into clinical trials and ultimately, obviously, FDA-approved drugs that you can actually get from your doctor? And so it's, I think it was a good way that Veronique sort of described it. Probably as many steps as there are letters in the alphabet. There may even be, depends on which alphabet you're talking about. But I think there are lots and lots of different steps along the way. So this is kind of a simplistic view, but it's worth kind of at least getting this in your mind as a concept. And it's really divided roughly into kind of 3 main areas. If you think about the basic side, this is again really where the scientists sit in their labs, uncovering sort of the secrets, sort of lifting up the car hood, if you will, looking at the engine, trying to figure out what's going wrong in Parkinson's disease, and coming up with insight about what might be ways to actually uncover new targets and new ways of thinking about Parkinson's. Once you've got those ideas, though, you actually have to turn them into something that you can actually treat people with. And so that's really where this sort of preclinical, or you may hear the word sometimes, translational space happens. And that's really about taking that idea and then turning it into an actual drug that you can start testing. And a lot of these may be small molecule chemical kinds of drugs, but we're also starting to see more and more some of these sort of very novel strategies, gene therapy, using stem cells, using a variety of different ways to actually treat a disease. And so this is really where this preclinical space is where a lot of that hard work happens. It's not sexy work. It's often sort of, just nitty-gritty kinds of work that isn't going to give you a particularly spectacular research paper in Nature or Science journals, which is what a lot of scientists like to have. But it's an area that is probably one of the most critical areas for actually getting drugs to people. Of course, then once you have a drug that seems to be safe, seems to hit on the mechanism you want to hit on, it then moves into the clinical testing phase. And that's obviously the most probably critical part of actually getting it to people, is we actually have to make sure it's safe and that it actually has some efficacy. And so we'll talk a little bit more about that at the end. In an ideal world, obviously this all is very straightforward. You go from one step to the next, and ultimately get to the clinic. In practice, though, what really happens is you learn along the way, you take a few steps back and rethink, go back to the drawing board a bit, and then you continue to kind of move down the way with more knowledge. And that's something that Carly mentioned that the PI here is really good about doing, because they're all in one place, so they can kind of learn along the way from the clinic, and they can immediately go back to Jeremy and say, Jeremy, you need to rethink this idea because we gotta move this forward. So it's a nice. It takes time. It's not an easy process, but it's there are a lot of different steps that can inform every step along the way. The problem, though, as we see it, is a lot of funding goes to basic research. A lot of this is from the government, from NIH. fairly far along in the clinical space, that's where really large, big pharma sits. They have deeper pockets and really can do those large sort of phase 3 efficacy trials that take millions and millions of dollars to run. But that middle ground, sort of the valley of death as we like to call it, that's a much harder space to really get a lot of movement on because there aren't a lot of people sort of constantly digging in that space, trying to help move those ideas forward. And so that's an area where the Fox Foundation, at least for Parkinson's disease, can really play a role. And that's really where we've excelled over the years in trying to sort of cherry pick those interesting ideas from the lab and actually move them along the critical steps to get them to the clinic. So before I move on, I think it's worth appreciating and understanding that we actually do have some treatments for Parkinson's today. And obviously, unlike other diseases that might not have any treatment options, we do have some ability to treat and manage the symptoms of Parkinson's. Most of this, though, is focused on a very sort of basic idea, which is about replacing the lost dopamine in your brain, which is obviously loss of dopamine is one of the key issues with Parkinson's disease. And so there are a number of different approaches that doctors can use now. They can either give you a sort of synthetic, essentially early version of dopamine that can be converted into the brain and become dopamine, and that's levodopa. Many of you are on Cinemet. That's essentially a version of levodopa. There are other kinds of dopamine replacement drugs that can target the different pathways that dopamine acts on in the brain as well, or keep dopamine around. For example, some of you may be taking Azilect or Rasagiline, which is the drug that Teva makes. That's another way to also kind of keep that dopamine around. So these are all fairly effective drugs. They help to kind of keep the dopamine running for a little bit longer, but they're by no means the cure. They're not going to save your dopamine neurons from dying off, and obviously the disease continues to progress. The other option that's sort of available to people now in the last 10 years or so is something called deep brain stimulation. Some of you may be aware of this already. Obviously, for later stage patients, when the drugs kind of stop working, they don't manage the symptoms as well. At least for some people, there is this option of getting deep brain stimulation. And so that is actually a big benefit for people, at least a subset of people who reach that stage. But independent of that, there's not much else out there that's really truly effective for people with Parkinson's disease. And that really then, I think, sets the stage for what we're trying to talk about today and really focus on. And that's how do we deal with a lot of these unmet needs that we need to do to actually improve treatments for Parkinson's. And so at the Fox Foundation, the way we sort of view this and strategize around it is really around sort of two main areas. And that's what can we do to help treat the symptoms of Parkinson's today? Also some of the side effects. Some of these drugs do have side effects, and if we can make those symptoms more treatable and we can make those side effects less cumbersome, we can actually make patient care today much better. The other side of that also is, though, independent of helping you sort of manage your existing symptoms, how do we actually alter the disease? How do we actually cure the disease at the end of the day? And so that's another big area that we're really focused on. So I'm going to talk about sort of examples of specific areas within each of these. So let's start with the hard one first, which is really how do we modify the disease, how do we alter the disease? And this is an area that the foundation from day one has been very much involved in. It's a huge part of our funding portfolio over the years. I think $270 million that we funded, a good $115 million of that has actually gone towards these disease-modifying efforts. And so the way we can think about that, this is really where, again, researchers like Jeremy can really help, because this is really trying to figure out what are the underlying causes of Parkinson's disease. What are the different areas in the body, the different mechanisms in our cells that we can actually target therapeutically? And this is about trying to really understand and uncover those. And so there are a lot of different areas that we can think about. We don't think of Parkinson's really as a genetic disease, but there are genetic forms of Parkinson's disease. And we can actually use those genetic insights to actually understand different pathways that in which Parkinson's could be treated. And so two good examples of that are in two genes, one called alpha-synuclein and one called LRRK2, and we'll talk a little bit more about that in a second. But that can really get us some really fundamental insight into the causes of Parkinson's and then actually build a drug development program around those ideas. But it's not the only area that we see a lot of excitement. I'll talk a little bit later about the area of trophic factors. So trophic factors, you can kind of think of it's like fertilizer for the brain. The idea here is we can actually put in your brain some chemical that can actually make those dopamine brain cells survive a little bit better, maybe even restore some of their function. And so that's a very active area of clinical research right now. But then there are a lot of other sort of biological pathways that people have been looking at as well. There's a growing amount of evidence to suggest that potentially one of the causes of Parkinson's, or contributors to Parkinson's disease might be brain inflammation. And so there's a lot of work trying to understand, can we lessen that inflammation and actually get some therapeutic benefit. I wanted to talk a little bit specifically about LRK2 because it's a very interesting example of how we can really focus on a potential cause of Parkinson's and really build a therapeutic program around it. And so for LRK2, this is a gene, mutations in LRK2 have been linked to Parkinson's disease in some families. Probably when you look at sort of the grand scheme of Parkinson's cases, anywhere around maybe 1 to 2% of cases are probably linked to a mutation in LRK2. So it's actually, when you look at the genetics, it's relatively one of the larger genetic contributors to Parkinson's right now. In particular, certain ethnic groups tend to have this mutation more frequently, Ashkenazi Jewish and North African Arab Berbers in particular. The biology of it is not well known, but one thing we do know about it is the kind of protein it is, the kind of enzyme it is in the cell is actually an enzyme that drug companies know how to make drugs against. And so there's been a lot of great movement in trying to understand not only LRK2's role in PD, but on the industry side, a lot of interest because it's a drug target they can actually work with. And so we've been working with a lot of our companies on this. So what we've been able to do at the Fox Foundation to really focus on a target like LRK2 is to really think about it in a broad strategy in 4 different areas that we can actually work in. So part of this is thinking about the biology of LRK2. How is it linked to Parkinson's? What goes wrong when you mutate this gene for LRK2? How does that actually impact the cells in our body? And we've developed a very large scale consortium of investigators, and I'll have Jeremy come up here in a second to talk about that, of investigators interested in understanding the biology around LRK2. In parallel to that, though, we can do a lot of other things as well. And so we've been developing a lot of research tools as well. This is something that scientists need in their labs to actually answer a lot of the questions that they have about LRK2. And so we can actually generate a lot of these tools ourselves from the Fox Foundation, make them either freely available or at very low cost, so that investigators like Jeremy don't have to use their entire research budget just to buy a couple of tools. And it really helps us to kind of grease the wheels, if you will, and keep research moving forward. We also are very interested in, like I said, working with companies. Since they are very interested in LRK2 as a target, we can work with them, understand the strategic decisions that they have to make, and try to work with them to make sure that we are prioritizing the right work in our efforts as well. And then finally, we obviously need to understand people with mutation. And so we've got, over the last 2 years, have developed a worldwide network of clinicians who see individuals with the LRK2 mutation, families with the LRK2 mutation in their family, and are bringing all those groups together to perform collaborative research efforts. And so it's a worldwide effort. I think we've got about 1,000 or more to maybe 2,000 individuals who carry the LRK2 mutation in that network now, and it's really becoming a really valuable resource for understanding LRK2-related Parkinson's disease. So I wanted to give Jeremy Nichols actually a chance. I been referring to him, but this is, I think, a really good example of an investigator we've been working with now a couple of years, where he's very interested, obviously, in LRK2, and we've been able to really help, I think, him in a variety of different ways, and I wanted to give him a chance to sort of talk about that.
So first, I just wanted to say, in this forum where we have people from the place that I work here at the Parkinson's Institute and with the Michael J. Fox Foundation, I just wanted to extend my thanks because I've been here for about 1.5 years. So in the entire time, I've essentially had funding from the Michael J. Fox Foundation, and this has not only facilitated my research, but it's actually accelerated it. And I'll try to touch on ways that's happened. And one of the reasons I bring it up in the beginning is because I'm going to let you know how the Michael J. Fox Foundation has been a portal for me to actually access multiple players in the field so that we can accelerate our research. And I think this plays into the theme of the day. So, one, Brian touched on a couple new targets for Parkinson's disease. And when I've been classically trained as a molecular biologist, a cell biologist, a biochemist, but not a person who deals directly with disease. And when I was a graduate student, when people wrote grants, they would have a small section of the public health importance. It was a necessary part of the grant, but not something that was actually fleshed out. And I think that over time, as the NIH monies became tight, those grants, those portions of the grant section became larger because they actually started to think about how they could become more relevant to helping people with disease. And I'll tell you, one of the first times when I started trying to define how my new research program was going to go, when I stopped being a trainee and being a program leader, it was actually at a Michael J. Fox Foundation meeting. At first I met Bill Langston there, which facilitated, I think, coming to the Institute. But also, Dave Weiner gave a talk, and it was it was essentially how we're going to define a clinical trial for a LRRK2-based therapeutic. And it was forcing me to step back and look at it in terms of how can my research not just keep me in a job, but how can I make what I do important for a drug company to take a molecule to the clinic. And it's not just thinking about the next research grant so that I can keep my 3 postdocs, but it's how can my next research grant get me 3 new postdocs to do some new research to move it forward, because my best ideas yesterday aren't my best ideas today. And that's one of the things the Fox Foundation does, is they have a very active interaction with the researchers, and one of them is through the LRRK2 Consortium. Every month I get on the phone with about 40 other researchers who are very impactful in their field, and we all share our stories. Now, this is going to lead into why we want to collaborate, or how we collaborate. And if you think about a young researcher who wants to build a research program and feed his wife and kids, you don't want to tell everybody your secrets, right? But if you want to make a difference in disease, you have to tell people your secrets. And you have to have a forum to do that, and I really thank the Fox Foundation because they've done that for me. And it's actually helped me do my research a little bit better. So when you collaborate, it's because you essentially have similar goals. And that's why I say if you have someone with a similar goal as you, you're not going to tell them how you're approaching that target, or the new tools that you're going to engage to get there. You're just going to keep that to yourself. But if we have a forum in which we can talk, and we can actually de-risk our investments, because we've already got an investment from the Michael J. Fox Foundation, we already have the funding to do some research. In my mind, that frees us up a little bit to work in a space where we can make a little bit of a difference. And so it's the common goals of the researchers that work on Parkinson's disease, and in particular targets, that the Fox Foundation has allowed us to come together in many ways. And one of the reasons you want to come together is that you should have a synergy. You don't want to come together with a 1 + 1 = 2. You want 1 + 1 = 4. And it's the ways you can do that is engaging different people with multiple different kinds of expertise. I've named a few expertise that I have, and I pretend I have more, but I have a set of skills that I think can complement other people's skills. And it's giving us the environment to engage those other people is where we're going to make the larger leaps instead of the baby steps. And so it's the more fruitful, multiple disciplinary approaches that are going to be successful. And that's one of the things that the Parkinson Institute has allowed me to do, because I can not only engage in basic science research, but I can talk with clinicians and say, how does this approach make sense? I can talk to somebody who designs clinical trials and say, what would be the best thing to do at this point in the trial? And so actually some of the funding from the Fox Foundation has been to facilitate our development of new technologies so we can assay these types of targets in patient samples. It's a real big difference from going in the clinic and trying to define the type of enzyme activity that something may have, to trying to understand that in a sample from a patient and how can we relate that to disease. And one of the things that we're doing with funding from the Fox Foundation is trying to develop an assay for LRRK2 so they can be applied in clinical trials so that companies can understand how efficacious their new compounds are. And this is one of the things where we're making leaps and jumps and not just baby steps. So the different kinds of places that you would interact with, I've mentioned intramural interactions here at the Parkinson's Institute. We have a grant from the California Institute for Regenerative Medicine, and this is a collaboration between Birgit Schule here at the Parkinson's Institute. She has a medical degree, but she has a large induced pluripotent stem cell program, and it only makes sense for us to apply our technologies to the new stem cell systems to understand Parkinson's. And also extramurally, I'll list a few other of my extramural collaborations. This is not an exhaustive review, but we've been able to engage other people at different institutes all across the world so we can try to make this research happen. And with industry, we've gotten, some of the grants that we've gotten have been with people in biotech. So we have for-profit companies working with non-profit companies to develop technologies so that we can start assessing how well these compounds work in a clinic. And being here at the Parkinson's Institute, we have a large degree of interest in pharma companies. It's because these pharma companies are actually developing the drugs that get into the clinic. For a long time, there's been a huge wall between academia and industry, and those walls need to come down before we make any progress. And if these companies are going to start making drugs towards these targets, we have to start trying to find a way to de-risk their programs. And that's one of the things the Fox Foundation does. They give money to pharmaceutical companies who need a little bit of cachet behind some of the work, or a little bit of de-risking their program, because it's a huge effort to try to engage a new target to make a drug to try to stop disease. And so you have to give them a little bit of leeway, and that's what the Fox Foundation does. But we've been able to also engage that because of our position here and our integrated approach to Parkinson's therapy. This allows us to occupy a unique position in the world because we can not only engage with pharma companies, but we can do it with basic science researchers and people that want to make money by developing technologies and also these drugs. It's by making this all funnel into one thing, and it's the end result is the patients. And I can tell you in this room every single day, I see Parkinson's patients not because I'm a physician, but because they come and they engage in the well-rounded Parkinson's Institute approach to how we treat people with disease, and that's because they have room to do exercise in and also voice classes and dance classes. And it's these things where you can have an approach where a scientist like me, who would typically be working at a biochemistry department, would not be engaging with clinicians nor seeing patients. I think this is the best place to be. And so I just wanted to wrap up by saying that we do have a lot of intramural collaborations, and we have a lot of reagent sharing and discussions that's actually facilitated through the Michael J. Fox Foundation. Every time we have a phone call, there's MTAs that fly around. And this is because people get on the phone and they tell each other what we're doing, and they say, I have this reagent. I want to, let you use it. Or they say, can I use your reagent? This is something that won't happen until after publication. And there's actually pharma companies involved in this consortium. So we have people that are actually making the drugs that are being engaged at this level. So we have basic scientists that for-profit pharma companies calling us, asking us for reagents. This is a fantastic divergence from previous ways to try to design drugs. We just were recently awarded a grant from the Fox Foundation to try to understand what happens to that molecule LRRK2 after you inhibit it with a drug that's been made by a pharma company. And this is with a collaborator that's in Belgium. This allows not just things that happen across the street, but across the world. And we have also just submitted another grant application in conjunction with a Bay Area pharmaceutical company to the Michael J. Fox Foundation, or PREAP. And what we've been able to do is to use the opportunities of the Fox Foundation to wrap in multiple different investigators with different expertise. So this company Elan up the road, and also a collaborator in Australia, we would like to, put a proposal together to do some work. These are opportunities that are only afforded to us by funding agencies like the Fox Foundation because they have a rapid response to investigators' questions and ideas. And with industry, we have open and active collaborations with Genentech and Life Technologies to develop assays for LRRK2. And we also are engaging small biotech companies that generate stem cell models for Parkinson's disease research. So we have multiple different ways that we can engage people, and I'll tell you that being able to work with the Fox Foundation and receive funding from them has allowed us to be not just successful, but I think we're moving the needle a little bit further. And that, honestly, they let me drive it till the wheels fall off. And that's what we need to do. We can't save this money. I mean, when I get money, I spend all of it. So, this is donor money, it's taxpayer money that we spend, and we've got to be good stewards of it. And the only way that I can be a good steward of it is if I realize what limitation I have and I can engage someone else so we can move that idea forward. So, I think that's about all I have.
So thanks, Jeremy. So I wanted to move on. We've been talking a lot about, obviously, altering the disease and ideas that are sort of in the more preclinical space, sort of ideas coming out of basic research labs. But it's important to actually step back and realize there actually are some ideas that are being tested in the clinic right now. And so there is a lot of bit of excitement in some of these approaches. I mentioned this is just sort of a short list. This isn't obviously comprehensive of every trial ongoing, but certainly I think some of the major trials that we are aware of and that the Fox Foundation is actually involved in. I mentioned Serogene trophic factors, so this is probably one of the more exciting trials ongoing right now. This is the idea of delivering, in this case, gene therapy for a trophic factor to the brains of people with Parkinson's, and actually seeing if that can rescue and maybe even restore a little bit of the function of in the brains of those individuals. And so that trial is ongoing. This is considered a phase 2 efficacy trial. I think we should have, hopefully, I think the last individual was recruited in that trial just recently, so we should hopefully have results probably in early 2013 or so. And so I think this is an area where we're really excited because this is an opportunity to really see if this idea works. But that's not the only trial that's ongoing. We actually have some other trials that are ongoing as well. I won't go through each one of these, but I wanted to note the one on the bottom. Afitope. This is a company of Fierus in Austria. They've got an interesting idea for targeting another genetic form of Parkinson's, alpha-synuclein. But what's interesting about alpha-synuclein is that everybody with Parkinson's disease actually has clumps of this alpha-synuclein protein show up in their brain. And so what Afiris is doing is they have essentially a vaccine approach to get rid of some of that clumping. And so this is an exciting area where we think we are finally starting to see sort of movement of a therapeutic for what we know is a fundamental potential cause of Parkinson's. That is now moving into the clinic, and that trial is just starting now. So definitely some areas to be excited about, because these are areas that obviously are in clinical testing now. In the next few years, we actually should see results from these. But I think that one of the challenges of doing disease-modifying trials is that it's not as easy as you might think. So the one problem with doing a disease-modifying trial is really at the end of the day, you're testing these individuals with this drug, and you're testing whether they're getting better. And the only real way we have of doing that right now is based on some fairly subjective clinical measures. You know, you go into the doctor's office, they ask you to do some of the tests that you guys all probably are aware of. You know, touch your nose, move your arms around, stand up, sit down, all those things that you do. There's a sort of a sophisticated scoring system that doctors use to try to basically apply a number to how severe your Parkinson's disease is. And it's pretty good at giving a general picture of the severity of your Parkinson's, but in a clinical trial setting, there's a lot of noise in that measure, and what can happen is it's going to be very difficult to know whether your drug is really having a true disease-modifying effect or not. And so what this means in practice is that trials have to be huge. You need lots of subjects to kind of get around that variability problem. It's a long process trying to sort of, detect this sort of potential disease-modifying benefit within sort of, again, that noise. And it can oftentimes, too, make interpreting those results very difficult, because you're not quite sure, was I just seeing sort of maybe a little bit of a symptomatic effect, not really a disease-modifying effect? And so it makes it really difficult to analyze these trials. And this really, I think, again, highlights the need that we have for a real biological measure of an objective measure of Parkinson's disease progression. And so this is something that the foundation has really made a big mission for us recently. Woody alluded to this early on about the Parkinson's Progression Marker Initiative, and this is a really large effort that we're involved in to try to identify this disease progression biological marker that researchers could actually use in clinical trials to determine whether the drugs are working.